All right. And when I say photoautotrophs, I mean eukaryotic photoautotrophs and prokaryotic photoautotrophs. So yeah, eukaryotic mein we have plants and we have algae, and prokaryotic mein we have the photosynthetic bacteria, for example, the cyanobacteria, which are also known as blue-green algae. So far, so good. ठीक. अच्छा. अब जितने भी photosynthetic eukaryotes हैं ना, whether it is plants or whether it is algae, they will carry out photosynthesis in an organelle known as the chloroplast, right? And इसमें यूकैरियोट्स सिर्फ इसलिए मैंने लिखा है बिकॉज इन प्रोकैरियोट्स जो बैक्टीरिया है, बैक्टीरिया में कोई मेम्ब्रेन बाउंड ऑर्गेनेल्स होते ही नहीं है, एंड क्लोरोप्लास्ट इज अ डबल मेम्ब्रेन बाउंड ऑर्गेनेल. देयरफॉर प्लांट्स में और एल्जी में ही सिर्फ क्लोरोप्लास्ट होंगे, एंड विच इज़ वाई दे विल conduct photosynthesis in the chloroplast. So yeah, so the chloroplast has two membranes. There's an inner membrane, there's an outer membrane. Okay, it has stacks of membranous structures called thylakoids. These stacks are called grana, singular granum, and each of these sacs is called a thylakoid. ठीक है, thylakoid के अंदर thylakoid का भी lumen होता है, and chloroplast में ये जो the empty void, this space is called the stroma. This entire space is called the stroma, which is not part of the membranous thylakoids or grana. This is the stroma, everything that's outside. ये तो बहुत basic structure है, which is also a continuation from your cell structure वाला chapter. Is that all right अभी तक? It's just revision. अच्छा. This is the very basic equation photosynthesis का. You take carbon dioxide in, you take water in, you take sunlight, and then you convert the carbon dioxide in the water to glucose. Glucose is the food, the intended product, and you also get oxygen, which in this context is simply a waste product. ठीक है, प्लांट को ऑक्सीजन का कोई काम नहीं है, उसको सिर्फ ग्लूकोज चाहिए. So glucose, food. Now photosynthesis has two stages. ठीक है, there is the light dependent reactions and then the light independent reactions. ठीक है, the light independent reactions will take place in the thylakoid membrane. तो ये जो thylakoid बना हुआ है ना, इसकी मेम्ब्रेन को हम जूम इन करके देखेंगे. दिस थिंग, वेट, ये कंफ्यूजिंग हो रहा है. टेक अ डिफरेंट शेड ऑफ ग्रीन. दिस, वी आर गोइंग टू जूम इन ऑन दिस. ये इसके अंदर हो रहा होगा, इसकी मेम्ब्रेन के अक्रॉस you will see the light reaction happening. Acha, one more thing. These are the inputs के water, sunlight, NADP plus and ADP. अब ये क्या है? These are this NADP plus is the electron carrier for photosynthesis. We'll come to this in a minute. Electron carrier for photosynthesis. And also one more thing that you should remember is the oxygen that we get in the light dependent reaction, the light dependent reaction, it comes from water and not from CO2. ठीक है, ये MCQs में पूछ लेते हैं, बहुत पूछते हैं कि ऑक्सीजन कहाँ से निकलती है. अभी तक does anyone have any issues so far? Okay. Yeah. So the purpose of the light reaction is to convert the energy of sunlight into cellular energy, ATP. Okay? So it takes the sun's energy and it converts it into the cell's energy. And in other words, you're making the energy. Now I know कि you can't make energy, लेकिन एक तरह से understanding के लिए हम कह सकते हैं कि हम energy जो cell जिस form में energy को use करता है, that is ATP. ठीक है? तो हम sun की energy को ATP में किस तरह से harness करते हैं, that is in the light reaction. That is what happens in the light reaction. और जो light independent reaction है, also known as the dark reaction, ठीक है, that simply uses the energy जो हमने light reaction में जो energy harness की, ठीक है, in the form of ATP, we're using that ATP to make glucose. That's what we're doing. So again, light independent reaction में क्या inputs हैं? You have NADPH and ATP. NADPH was formed in the light reaction. ATP also formed in the light reaction, and CO2 is something जो आती ही light independent reaction में, light reaction में CO2 का कोई काम नहीं होता. This is something that जो Calvin cycle में CO2 enter होती है later. And the site of the light independent reaction is the stroma. ठीक है? Now you come outside the thylakoid membrane and you enter the stroma. ठीक है? Outputs इसके NADP plus NADPH जो हमने इलेक्ट्रॉन कैरियर लिया था वहाँ पे NADP plus वो NADPH में कन्वर्ट हुआ था लाइट रिएक्शन में. अब वो वापस रिसाइकिल होके, because this needs to be recycled, this is an electron carrier. Again, not much importance, but still good to know. So let's start with the light reactions officially. Again, this is the energy capture phase of photosynthesis. वही वाली बात, you're taking the sun's energy and you're going to convert it into a form that the cell can use. And the main function is to produce ATP and NADPH, electron carrier of photosynthesis. Acha, do not confuse this with NADH, because NADH is the electron carrier in respiration. NADPH is the electron carrier of photosynthesis. Any questions so far? Anything that was, problematic? Saya. So I'll explain this when we do the Z scheme wali cheese. So energy from sunlight is captured by molecules called pigments. So yeah, अब जो pigments, the pigments that we're concerned with, that's chlorophyll A, chlorophyll B, and carotenoids. Ab chlorophyll A and chlorophyll B, they will be absorbing all the shades of light except green. इसीलिए हमें chlorophyll green दिखते हैं. और carotenoids जो होंगे, they will absorb these colors, green, blue, indigo, violet. इसीलिए they appear reddish and orangish. अब ये जो specifics है ना कि कौन सा pigment कौन सी light absorb करता है and what colors they appear, this is something that you should cover from the notes in the book, क्योंकि ये फिर factual recall type questions में ये आ जाता है. So I would advise you to refer to your textbooks for that. Acha, what do you want me to repeat? The pigments part? Muskan? The first one, starting from the beginning, photosynthesis का जो overview दिया था मैंने, that, did you join late? Just let me know in the chat exactly what's bothering. Before pigments, अच्छा, let's see. Before pigments मैंने ये कहा था कि हम light dependent reactions का जो main purpose है, that is to get ATP and that is to get NADPH. सही है? NADPH बनता है from NADP+. ठीक है? This is an electron carrier. और ये electron carrier है photosynthesis का. ये repeat करना था, मुस्कान? Is that what you missed? आई थिंक अब क्लियर होगा. राइट. So this is again, अच्छा, we know from physics के light is carried on a photon. Light जो होता है, um, light travels as packets. ठीक है, there are packets called photons, energy packets, and light also travels as waves, and it's part of the electromagnetic spectrum. ठीक है, and we know कि जितनी भी high frequency waves हैं, they will have shorter wavelengths and they will carry higher energy. And we know that visible light itself, it's made up of 7 colors which have a certain range. ठीक है, 750. अब ये जो है ना, in order of decreasing wavelength है, decreasing wavelength है. अच्छा, अब ये 380 and 750 बहुत specific numbers हैं. Again, you should read from your book. ये book में क्या number दिया हुआ है. A safe range to remember is 400 to 700 nanometers, जो visible light की range है. This is a safe range to remember. And if we were to zoom in on the thylakoid membrane, this is what we would see. You would see something called a photosystem. You would see uh, the cytochrome complex and the electron carriers and the NADP plus reductase, which we're going to discuss abhi. ठीक है. अच्छा, so before we begin, I want to explain these terms, the photosystem and the reaction center and the light harvesting complex. ठीक है. A photosystem is a reaction center plus the light harvesting complexes. The reaction center simply consists of a pair of chlorophyll A molecules and the primary electron acceptor. And the light harvesting complex is simply pigment molecules and proteins. Okay? The reason I wrote it in this way is because I think in the Sindh wali book, ये चीज़ें ये terms mentioned हैं, but they're not explained in a lot of detail. तो you should know कि exactly हर चीज़ है क्या ना. Reaction center है, तो reaction center किस चीज़ का बना हुआ है. It has 2 molecules of chlorophyll A and a primary electron acceptor. Now the photosystem, हम किस चीज़ को बोल रहे हैं. Photosystem would be everything. ये chlorophyll A and the primary electron acceptor and the light harvesting complexes. ये इन सब का जो, if you were to group these things together, that is what is one photosystem, the PS1 and the PS2. It's simply, in even more simpler words, it's simply the pigment molecules and proteins associated together to form a photosystem. अभी I know ये जरा सा segmented लग रहा है, electron carrier समझना है. हाँ ठीक है, I'll explain it. अभी जब हम when I'll officially like begin the Z scheme और ये सब, तो electron carriers वाली चीज़ समझ आ जाएगी hopefully. हाँ, now let's begin. अब what happens is कि we know that the sun is the ultimate source of energy, right? जितनी energy आ रही है वो sun से आ रही है. ठीक है, तो ये जो sun की जो energy है, that is captured by the pigment molecules. ठीक है, ये photosystem 2 है, and photosystem 2 appears before photosystem 1 क्योंकि वो डिस्कवरी पहले हुई थी photosystem 2 की, so that's the order in which we study. So what happens is that when light energy falls on a pigment molecule, so the pigment molecule gets excited. Now I'm not saying chlorophyll or carotenoid or whatever, I'm just saying pigment क्योंकि photosystem के अंदर बहुत सारे pigments हैं. Reaction center के अंदर chlorophyll A है. Make sense? Electron acceptor पे आते हैं अभी. Right. So any pigment molecule जिस पे light पड़ती है, that gets excited, and when it gets excited, it releases electrons, and then those electrons, they are transferred from pigment to pigment to pigment molecule until they reach the reaction center. अब ये reaction center है, ठीक है. यहाँ पे you have your chlorophyll A pair, and ये जो फोटो सिस्टम है PS2, इट इज़ ऑल्सो कॉल्ड P680, क्योंकि इसका जो क्लोरोफिल ए पेयर है ना, दैट अब्जॉर्ब्स अ वेवलेंथ of 680 nanometers. इसीलिए it is called P680. PS2 is also called P680. That is just a fact that you need to know. I'll add a star next to it. हाँ, so back to this. यहाँ पे you have your chlorophyll A pair. अब ये chlorophyll A pair पे जब finally electrons जाते हैं तो ये भी excite होते हैं. Now when they get excited, the electrons will finally travel through the chlorophyll A pair. ये भी अपने electrons lose करेगा और ये देगा एक primary electron acceptor को. Now this is simply a molecule. This primary electron acceptor does exactly that. इसके नाम में लिखा हुआ है primary electron acceptor. ये वो molecule है जो chlorophyll A pair से chlorophyll A वाले जो molecules हैं उनसे सबसे पहले electrons लेगा. That is why it is called primary. And again it accepts electrons. ठीक है, तो photosystem 2 का जो electron acceptor है, that is called pheophytin. That is again a name that you need to know for the exam, because ये पूछते हैं. तो now what this pheophytin does is, इसके पास भी electrons आए, अब ये अपने electrons किसी और चीज को देगा. ये अपने electrons देगा to a series of compounds which make up a chain. ठीक है, they make up a chain and it's called the electron transport chain for very obvious reasons, क्योंकि ये all they're doing so far is transporting electrons. जैसे एक मेल डिलीवरी सिस्टम होता है ना, कि पॉइंट ए से पॉइंट बी, फिर पॉइंट बी से पॉइंट सी, करते 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 अपने इंटेंडेड रेसिपिएंट तक पहुंचता है. तो इसी तरह से इलेक्ट्रॉन्स पहुंच रहे हैं. ठीक है, तो यहाँ पे लेट्स से वी पोस्टेड द, हमने अपना जो भी पार्सल था वो हमने ड्रॉप किया टू फेडेक्स और वेरेवर, टू अ कोरियर कंपनी. अब वो मल्टीपल 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 एक चेन है पूरा. वो पार्सल यहाँ जाएगा, फिर यहाँ जाएगा, यहाँ जाएगा, यहाँ जाएगा, यहाँ जाएगा, एंड इवेंचुअली इट विल रीच इट्स इंटेंडेड टारगेट. और उसका जो इंटेंडेड टारगेट है वो है ये NADP प्लस रिडक्टेज. सॉरी, दैट्स द टारगेट, दैट्स द रेसिपिएंट जिसके पास जाने हैं इलेक्ट्रॉन्स. ठीक है, I hope your question was answered. What is an electron acceptor? Any questions? ठीक है. So yeah, अब होगा क्या, जो प्राइमरी इलेक्ट्रॉन एक्सेप्टर है which is pheophytin, it will transfer electrons to plastoquinone. This is plastoquinone. इसका short form है P. Okay, I'll just write it down. PQ is plastoquinone. And then you have something called the cytochrome complex. It is also called cytochrome B6 something ऐसे. And then C here stands for plastocyanin. That's another compound. Now these are names that you should know. ठीक है, प्लास्टोसाइनिन. अब क्या हो रहा है, यहाँ से हमारा क्लोरोफिल ए मॉलिक्यूल जो था वो एक्साइट हुआ, उसने इलेक्ट्रॉन्स लूज किए. वो इलेक्ट्रॉन्स कहाँ गए? प्राइमरी इलेक्ट्रॉन एक्सेप्टर, ऑल्सो नोन एज pheophytin. Now pheophytin throws the electrons to plastoquinone, cytochrome B complex, and then plastocyanin. Now what has happened is कि ये वाला, ये जो हमारा क्लोरोफिल ए मॉलिक्यूल है ना, इट इज डेफिशिएंट इन इलेक्ट्रॉन्स. क्यों? क्योंकि इसके इलेक्ट्रॉन्स निकल गए. अब ये अपने इलेक्ट्रॉन्स कहाँ से पूरे करेगा? बिकॉज अगेन ये इट्स नॉट अ वन टाइम थिंग. लाइट तो इस पर फॉल करती रहेगी ना, इस तरह से अपने इलेक्ट्रॉन्स ये देता तो नहीं रहेगा. इट नीड्स टू ऑल्सो फुलफिल इट्स डेफिसिट. अब ये अपना डेफिसिट यहाँ से पूरा करेगा वाटर से. जो हमने वाटर, द वाटर दैट अपीयर्स इन आवर इक्वेशन, जो लाइट रिएक्शन में हम यूज़ करते हैं, वो वाटर अब यहाँ पे काम आएगा. So light is used to break down water molecule. Now this is called photolysis. Okay? Because you're using light to break down water. Lysis हो रहा है. So photolysis. And water gets broken up into hydrogen ions and oxygen gas. यहाँ पे oxygen gas भी इसी तरह release हो रही है. Oxygen gas निकल रही है in this process. Photolysis की वजह से oxygen निकलती है as a waste product. So जो इसकी electron deficiency थी वो पूरी हो गई by the photolysis of water. Moving on. So electron transport chain से eventually it reaches the electrons that were lost from photosystem 2, they reach photosystem 1. Photosystem 1 में plastocyanin से ये जाता है photosystem 1 के reaction center में. Electron photosystem 1 के reaction center में जाएगा. यहाँ पे भी chlorophyll A molecules हैं. And because these chlorophyll A molecules will absorb light of 700 nanometers, तो इसीलिए इस photosystem को P700 कहते हैं. Again, need to remember this. And ये अपने electrons फिर lose करेगा. इसका जो primary electron acceptor है, इसका कोई नाम दिया हुआ नहीं है in the books. So just remember this as the primary electron acceptor of PS2. ठीक है? इसका अब जैसे कि इसका नाम है ना फियोफाइटिन, PS2 के प्राइमरी इलेक्ट्रॉन एक्सेप्टर का नाम नहीं दिया हुआ इन द बुक. So far any questions, any confusion? ठीक है. या सो अब ये भी प्राइमरी इलेक्ट्रॉन एक्सेप्टर इलेक्ट्रॉन एक्सेप्ट करेगा एंड देन इट विल transfer the electron along a second electron transport chain. यहाँ पे you have a protein called, not a protein, a compound, it's called ferrodoxin. Ferrodoxin, ठीक है, FD. Ferrodoxin फिर finally electrons दे देगा to the intended recipient which is NADP plus reductase. अब ये एक एंजाइम है, ये कुछ नहीं करेगा. ये NADP plus जो इलेक्ट्रॉन कैरियर है ना उसको लेगा और इसको कन्वर्ट कर देगा NADPH में. NADPH की इंपॉर्टेंस है इन द नेक्स्ट स्टेप इन द कैल्विन साइकिल. एंड वेट, एक्सप्लेन कहाँ पे. हाँ यहाँ पे. तो NADP plus NADP reductase क्या कर रहा है? NADP reductase had NADP plus. ठीक है, ये if you know your chemistry, जो redox reactions वाला जो principle है, can you tell me कि NADP plus इस वक्त oxidized form में है या reduced form में है? NADP plus oxidized है या reduced है? To begin with, do we have the version of the compound that is oxidized or reduced? Oxidized. Yes, it is oxidized because इस वक्त it doesn't have electrons. इसके पास excess नहीं है अभी electrons का. उसके वो it has a deficit of electrons. Yeah, oxidized. But now what we'll do is कि हम NADP plus को force करेंगे कि वो electrons accept करें and become something else. So NADP plus is reduced to NADPH. ठीक है, so we start with NADP plus. इसमें एक hydrogen ion भी जाता है. It takes a hydrogen ion and it becomes NADPH2 plus. Then you give it one electron, it becomes NADPH plus. You give it another electron, it becomes NADPH. इस मेन चीज ये याद रखने की है कि ये NADPH बन जाता है NADP plus reductase की एक्टिविटी से. NADP plus reductase. एंड का जो स्टेप है. Any questions so far? I hope all that was clear. So yeah. Up, after this entire explanation of the one part of the light reaction, अभी non-cyclic भी रहता है. So the main purpose is कि जो it is actually electron flow which is contributing to this conversion of sunlight into ATP. ठीक है, of solar energy into cellular energy. ठीक है, electron flow की वजह से हो रहा है. Now this electron flow is of two types. You have linear electron flow या फिर non-cyclic, and then you have cyclic. अब non-cyclic में क्या हो, अभी हमने जो किया that is non-cyclic. It is linear. And why is it linear? Because जो electrons इसने lose किए क्लोरोफिल ए ने और फोटोसिस्टम 2, ये इलेक्ट्रॉन्स उसने दे दिए अपने. ठीक है, वो इलेक्ट्रॉन्स ट्रैवल किए, दे ट्रैवल लाइक दिस इन अ लिनियर फ्लो. इस तरह से इट ट्रैवल्ड यहाँ से यहाँ तक टू द प्राइमरी इलेक्ट्रॉन एक्सेप्टर, देन द चेन, एंड देन यहाँ से इट्स लाइक जो जेड बन रहा है ना, द वेरी फेमस जेड स्कीम. ठीक है, दिस इज लिनियर. इसमें जो इलेक्ट्रॉन्स इसने लूज कर दिए वो लूज कर दिए, उसको उसके पास वापस नहीं आए वो इलेक्ट्रॉन्स. इट्स नॉट लाइक दोज इलेक्ट्रॉन्स ट्रैवल्ड along this pathway and then they came back to photosystem 2. That did not happen in her. What happened was कि उसने अपने इलेक्ट्रॉन्स रीगेन कर लिए, but it regained them from another source. It's like you know when you lose your favorite toy as a kid, so your parents buy you a new one. It's not like you lost. If you were to find it the next day, that would be cyclic. But if you were to not find it and you got a new one in its place, that is non-cyclic. I hope that example makes sense. Great. Yeah, so I was saying कि non-cyclic और cyclic electron flow में जो difference है वो क्या है. So I was explaining, wait a minute. I was explaining कि जब photosystem 2 ने अपने electrons lose किए, तो they traveled in a linear sequence. They went यहाँ से यहाँ, and then they went along the electron transport chain, and finally they were accepted by NADP plus reductase. ठीक है, वो electrons वापस नहीं आए to photosystem 2. ठीक है, those same electrons did not go back to photosystem 2. उसके पास जो डेफिसिट पूरा हुआ वो एक एक्सटर्नल सोर्स से पूरा हुआ, एंड दैट एक्सटर्नल सोर्स वाज द फोटोलिसिस ऑफ वाटर. एंड सो आई वाज सेइंग कि जो छोटे बच्चे व्हेन दे लूज देयर फेवरेट टॉय से एंड देन दे गेट अपसेट कि हमारा टॉय हमारा ले लिया, तो अगर उनको अपना टॉय वापस मिल जाए, दैट इज साइक्लिक, क्योंकि उनको वही चीज़ वापस मिल गई. ठीक है, वो गई होगी, इधर उधर हुई होगी, बट देन वापस उन्हीं तक आ गई. बट अगर उनको ना मिले, then their parents have to buy them something new to replace the deficit. That is linear electron flow या non-cyclic electron flow जैसे दिया हुआ है बुक में. तो non-cyclic में आपको वही चीज़ वापस नहीं मिलती, उसका कोई alternative मिल जाता है. And cyclic में you get the same thing back. We're going to do cyclic in a moment, so that will be clearer. Whoever wanted me to repeat, I hope their question has been answered. Alize? Great. Yeah. Now we're going to do cyclic electron flow. अब cyclic electron flow में क्या होता है कि जो electrons photosystem 1 lose करेगा. अच्छा, so to begin with, यहाँ पे non-cyclic में क्या हो रहा था? Non-cyclic में दोनों photosystems were being involved. Okay, photosystem 2 lost its electron, एंड देन इवेंचुअली वो यहाँ तक पहुंचा फोटोसिस्टम 1 में. फोटोसिस्टम 1 लॉस्ट इट्स इलेक्ट्रॉन, ठीक है, बट देन इट रीगेन्ड इट्स इलेक्ट्रॉन एज वेल. वहाँ उसको उसने वहाँ से रीगेन किया ये इलेक्ट्रॉन, उसने अपना इलेक्ट्रॉन यहाँ से लूज किया, और फिर जो इलेक्ट्रॉन ट्रांसपोर्ट चेन से इलेक्ट्रॉन आया वो उसने उसका डेफिसिट पूरा कर दिया. दैट इज व्हाट वाज हैपनिंग इन लिनियर इलेक्ट्रॉन फ्लो, नॉन साइक्लिक में. नॉन साइक्लिक में फोटोसिस्टम 1 भी इन्वॉल्व हो रहा है और फोटोसिस्टम 2 भी इन्वॉल्व हो रहा है और 1 भी इन्वॉल्व हो रहा है. नाउ साइक्लिक में क्या होगा, यू विल जस्ट सी फोटोसिस्टम 1. अब ये जो नहीं है ना, अब ये जो रिमेनिंग पार्ट ऑफ द जेड स्कीम दैट हैज बिन सॉर्ट ऑफ ग्रेड आउट टू इंडिकेट कि इट इज नॉट पार्ट ऑफ साइक्लिक इलेक्ट्रॉन फ्लो. ठीक है, ये सब फोटोसिस्टम 2 इज नॉट अ पार्ट, इट्स नॉट इन्वॉल्व. ठीक है, लाइकवाइज ये इसका प्राइमरी इलेक्ट्रॉन एक्सेप्टर, ये प्लास्टोक्विनोन, ये सब ये नहीं चाहिए. यहाँ पे जो हमारे इंटेंडेड रेसिपिएंट है ना, वो भी यहाँ से गायब है, क्योंकि हमें NADPH नहीं बनेगा अब यहाँ पे. यू विल सी समथिंग इंटायरली डिफरेंट हैपनिंग. अब होगा ये कि यहाँ पे द स्टार ऑफ द शो इज फोटोसिस्टम 1. ठीक है, यहाँ पे फोटोसिस्टम 1 अपना इलेक्ट्रॉन लूज करेगा. इट विल, the electron will be captured by the primary electron acceptor which is unnamed. ठीक है, primary electron acceptor will transfer the electron to ferredoxin. Ferredoxin will transfer to the cytochrome complex plastocyanin. तो it's a cycle. This is cyclic flow. अब इस cyclic flow के अंदर NADP has no role at all. NADP plus has no role at all. So NADPH is not formed. And that is like the key difference between cyclic and non-cyclic of electron flow. ठीक है, अब यहाँ पे one thing that I didn't mention was ATP synthesis. The main चीज जो हमें करनी है, हमें तो ATP भी चाहिए ना, energy चाहिए for the next step. वो energy कैसे आएगी? Now what happens is कि as all these electrons are being shuttled from place to place along the electron transport chain, तो ये जो transfer of electrons है, now this is a series of redox reactions. ठीक है, you lose an electron, you gain an electron, you lose an electron, you gain an electron. That's a series of redox reactions. अब यहाँ पे energy is involved, energy is involved. अब energy is released एक तरह से, just every time an electron is lost, energy is released. अब ये energy is used to drive the synthesis of ATP. अब ATP बनेगा by another by a completely different process, that is chemiosmosis, which we'll discuss in a minute. ठीक है, so यहाँ से the key point to remember is कि electrons जब flow करेंगे तो electron flow की वजह से ATP synthesize होगा. I hope that is clear. Any questions? Okay. Yeah. So these are the key differences between cyclic and non-cyclic, and you can read this from the notes. ये notes का ही snapshot है. So chemiosmosis, that process that I was talking about. I'm looking for that diagram that I showed in the beginning. Thylakoid membrane वाला, ये रहा. Yeah. तो यहाँ पे what will happen is कि जो हाइड्रोजन आयंस निकल रहे थे ना फोटोलिसिस ऑफ वाटर से, तो just give me 2 minutes please. Yeah. Guys, give me 2 minutes. All right. Yeah. So I hope my screen is visible. हाँ, सही है, नजर आ रहा है. ठीक है. So chemiosmosis. Now what happens is we pump hydrogen ions from the stroma into the thylakoid space. अब stroma कहाँ है इसका? This is the stroma of the chloroplast. यहाँ पे hydrogen ions कम है. We use a hydrogen ion pump, ठीक है, to pump hydrogen ions from the stroma into the thylakoid space. Now remember कि यहाँ पे तो hydrogen ions अभी बन रहे हैं because of the photolysis of water. तो वैसे तो hydrogen ions यहाँ पे ज़्यादा होने चाहिए, लेकिन हम बाहर से अंदर की तरफ पंप कर रहे हैं. स्ट्रोमा में कम hydrogen ions है, but we're pumping those less hydrogen ions into the thylakoid space. ठीक है, using our hydrogen ion pump. This is the pump, it's not labeled. और यहाँ पे ATP synthase enzyme भी उसी के साथ है, attached है. So we pump hydrogen ions inside, and what we're doing is we're building an electrochemical gradient. सही है. अब hydrogen ions are charged, इसीलिए electrical gradient भी है, and chemical gradient, obviously because of the difference in the concentration. ठीक है. तो hydrogen ions हम वहाँ पे pump कर रहे हैं, हम वहाँ पे भर रहे हैं. Now we're pumping the ions and we're creating a strong electrochemical gradient. We're creating a difference in concentration. Obviously this, अब यहाँ पे एक potential होगा ना, एक potential generate होगा. Now there's these hydrogen ions will want to break free. They will want to break free. जैसे ही उनको मौका मिले, they'll rush out. So what happens is कि एक channel protein खुलेगा, ठीक है. Never mind what that channel protein is, just so that you can go with the flow. We're pumping hydrogen ions, and then those hydrogen ions will want to come out, and as they come out into the stroma, तो they will rush out. जैसे एक waterfall होता है ना, like when water falls from a very great height, तो बहुत energy के साथ flow करता है. तो इसीलिए जब hydrogen ions एक बहुत high potential के साथ वापस निकलेंगे ना towards the stroma, तो they will rush out with a lot of energy, and then that energy will be used to drive the synthesis of ATP, the conversion of ADP. ठीक है, ADP में phosphate लग जाएगा, एक high energy bond बनेगा, and you'll get ATP as a result of chemiosmosis. That's like a very big picture overview. Iska. Pumping of hydrogen from stroma to the thylakoid lumen is an active process. Yes, exactly, it's an active process. Pumping is an active process. Okay. Any questions so far about this? We discussed के NADPH कैसे बना in the process of the light reaction. We discussed non-cyclic electron flow. We discussed cyclic electron flow, and we also discussed the synthesis of ATP. अभी तक any issues? Okay, so all I'm saying is कि hydrogen ions यहाँ पे accumulate होंगे, right? Hydrogen ions are charged particles, and then that will create a strong electrochemical gradient. ठीक है, एक concentration gradient होगा, एक electrical gradient होगा. अब obviously the minute they get their chance, they will want to flow out, and when they will flow out, they will flow out with a lot of energy. बहुत energy के साथ निकलेंगे, and so when they will rush out, so that energy will be used to convert an ADP molecule into a high energy ATP molecule. That's all I'm saying. There's a proper explanation. Let me show it to you, and hopefully that will make it clearer. Yeah. The energy of the electrons is first used to pump hydrogen ions from the stroma into the thylakoid space. सही है. तो जो हम pump कर रहे हैं ना, someone said it is an active process. It is an active process that consumes energy. तो वो energy कहाँ से आ रही है? वो energy electron से आ रही है. You pump hydrogen ions from the stroma into the inner thylakoid space. This creates a strong electrochemical concentration gradient. The gradient contains stored potential energy. Next, a channel in the thylakoid membrane will open and electrons flow back into the stroma down their concentration gradient. The channel will contain the ATP synthase complex. which will use this energy to synthesize ATP from ADP. It's written in proper words. So I hope that explains it. Alize, does anyone else have any questions? यहाँ से ठीक है, understood. तो ये लाइट डिपेंडेंट रिएक्शंस तो हो गए. नाउ लेट्स मूव ऑन टू लाइट इंडिपेंडेंट रिएक्शंस कि अब जो एटीपी बना और जो NADPH बना वो कैसे बनेगा, ग्लूकोज कैसे बनेगा उससे. So I'll show you the pre-med notes. Where did they go? I think someone is trying to join. Pardon it all. Okay. Yeah. So the light independent reactions are also known as the dark reactions, and it is said that this is not the right term to use. कि you cannot say कि ये dark reactions हैं क्योंकि ये dark में नहीं होते. ये they can also happen during daytime. So they do not require the energy of sunlight, इसीलिए they're called dark reactions. Okay? They were discovered by Melvin Calvin and his colleagues in 1950. Factual recall, please do learn this. Where does it take place? It takes place in the stroma of the chloroplast, and it is also called the Calvin-Benson cycle. It's also called the C3 cycle. The C3 cycle as well. अब सबसे पहले तो you need to remember the names of these three stages: carbon fixation, reduction, regeneration. I'll show you the diagrammatic representation. अब in this phase, what will happen is कि हमारे पास क्या products आए वहाँ से? We got ATP, we got NADPH, assuming that this was non-cyclic electron flow, and then that's what we get from the light reactions. Now we will get CO2, और CO2 अब enter करेगा. And so what happens is CO2 enters through the stomata, enters the leaf through the stomata, and it is captured by an enzyme called Rubisco. Okay, now this is an enzyme, and I think it is supposed to be the most abundant enzyme. Acha, has anyone come across this? Rubisco is the most abundant enzyme. Anyone doing questions ya kahin bhi? Because I remember something along those lines. है ना? I'll still verify this from the book and then I'll add it to the notes. It is the most abundant enzyme. So what Rubisco does is it will convert this carbon dioxide. Carbon dioxide is simply, one carbon molecule. ये 3 carbon dioxide molecules lega, and it will create a short-lived intermediate compound. Wait a second. It will create 3 molecules of this compound. Let's count how many carbons it has. 6. Yeah. So this is a 6 carbon compound. And then वो compound टूट के it will become 3-phosphoglycerate. 3-phosphoglycerate will be further phosphorylated by the addition of ATP, and it will become 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate will be यहाँ पे NADPH आके अपने इलेक्ट्रॉन्स दोबारा लूज करेगा टू बिकम NADP+, ठीक है. एंड देन यहाँ पे रिडक्शन हो रही है. दिस फेज इज कॉल्ड रिडक्शन. व्हाई? बिकॉज NADPH इज कमिंग एंड इट विल रिड्यूस दिस कंपाउंड. ये जो कंपाउंड है दिस इज बीइंग रिड्यूस्ड. 1,3-bisphosphoglycerate. It is being reduced to glyceraldehyde 3-phosphate, also known as PGAL or phosphoglyceraldehyde. And then phosphoglyceraldehyde is the output basically. ये 3-carbon compound है, 3-carbon sugar है, and then 3 ये phosphoglyceraldehyde से it will be converted into glucose and other organic compounds. But that's not it. अभी हमें regenerate करना है. This compound RUBP that we started with. We need to regenerate ribulose 1,5-bisphosphate. And so what we will do is कि यहाँ से एक कार्बन निकल गया. Now you have your 5-carbon compound remaining, and then that will be regenerated by the addition of ATP. There is a better diagram that I should show you for this. So yeah, the Calvin cycle starts when ribulose 1,5-bisphosphate, a 5-carbon sugar already present in the stroma, reacts with CO2 to form a 6-carbon compound. This compound is unstable. It splits up into 2 molecules of 3-phosphoglycerate, and the enzyme Rubisco, regarded as the most common protein in nature. Yeah, so I was right. Is the most common protein in nature. RUBP molecules are regenerated so as to continue the cycle. With the regeneration of RUBP, the Calvin cycle or the dark reaction is complete. अब इसमें very important thing to remember is the number of molecules that are being, you need to keep track of the number of compounds, ठीक है, number of molecules of each compound. So इसमें you should know कि with each turn of the Calvin cycle, ठीक है, it's a cycle, right? So it's going to run a certain number of times. एक दफा चलेगा, फिर दूसरी दफा चलेगा. So that's one turn of the cycle, ठीक है. So with one turn of the cycle, one carbon dioxide molecules is fixed into RUBP. RUBP is a 5-carbon compound, ठीक है. ये 5-carbon compound CO2 के साथ जाके जब react करेगा plus 1 carbon, you will get a 6-carbon compound. वो unstable था तो वो टूट गया into 3-carbon compound. So 3 turns of the cycle correspond to the formation of 1 molecule of phosphoglyceraldehyde. 3 turns of the cycle. और अगर हमें 1 molecule of glucose चाहिए तो we need to run the Calvin cycle 6 times. And for 1 turn, 2 ATPs. Okay, 2 ATPs are used in the reduction process, and 1 ATP for regeneration. So that means okay, 3 ATP molecules in total are consumed. and in one turn, 2 NADPH molecules. Reduction. Now the reason why you should know this is कि sometimes they can give you questions जहाँ पे कोई ना कोई calculation involved हो, जहाँ पे they might ask you कि एक glucose molecule बनाने में कितने ATPs और कितने NADPH consume होने चाहिए. Can anyone tell me? अच्छा, first tell me if this is clear. Does anyone have questions इसमें से from the Calvin cycle or the calculation? No questions? Acha, so assuming कि in one turn of the Calvin cycle, we utilize 3 ATP and 2 NADPH. तो अगर हमें एक phosphoglyceraldehyde का molecule चाहिए, so how many times should the Calvin cycle run? 3 times. And if I were to ask you how many ATP molecules are consumed to form one molecule of PGAL, 9 exactly. So बस यही है, if you keep this basic number in mind, तो फिर calculations करना आसान होगा. Because what happens is कि अगर आपने ये चीज़ revise करके आप नहीं गए हुए होते हैं, तो in the अगर question आ जाता है, then it can be very confusing. You're trying to recall all the steps, and then आपने instead of writing 2 NADPH, आपने 3 कर दिया, calculation is going to be messed up. So there are past paper questions which I'll assign as homework, so that you can practice this concept. अच्छा, one more thing, ये जो PGAL है, इसके there are 3 alternative names. We call this phosphoglyceraldehyde, ये तो हो गया था. We call this G3P, glyceraldehyde 3 phosphate, and we also call this triose phosphate. Triose again 3 carbon sugar, इसलिए 3 carbon to triose. So that's the Calvin cycle done, and that's photosynthesis done. Now we can move on to respiration. ये page delete कर देते हैं. Okay, so we do have a small subtopic. This was the Calvin cycle, but there are alternative mechanisms of carbon fixation as well. अब ये जो process था कि the process of CO2 combining with RUBP, that is called carbon fixation. Okay. So the Calvin cycle is one way that carbon dioxide gets fixed into RUBP. But there are other mechanisms as well. क्यों? Because different plants have different environmental requirements. And mainly because जो हमारा enzyme है ना Rubisco, इसकी एक खराबी है. And the problem with Rubisco is कि it likes to combine with oxygen as well. Can bind oxygen. हमें oxygen से bind नहीं कराना. अगर हम oxygen से bind कराएंगे, तो we will have a very wasteful process called photorespiration. Oxygen. This we have to avoid because it is wasteful. So, on hot, dry days, C3 plants close their stomata, conserving water but keeping CO2 out and oxygen in. Under these conditions, photorespiration can occur. Rubisco binds oxygen instead of CO2, which consumes ATP and releases CO2 without producing ATP or carbohydrate. अब इसमें फायदा नहीं हो रहा ना प्लांट को, because what it is doing is it is using ATP without any reason. CO2 बाहर निकल रही है और कार्बोहाइड्रेट भी नहीं बन रहा. So this is why photorespiration is wasteful. It's a waste. We don't want this to happen. So some plants have adaptations. So there are different types of plants depending on how they do carbon fixation. C4 plants जो होते हैं, they minimize the cost of photorespiration by incorporating CO2 into 4 carbon compounds. 4 carbon compounds, इसीलिए इनको C4 भी कहते हैं, in mesophyll cells. These compounds are exported to bundle sheath cells, just remember the name, where they release carbon dioxide for use in the Calvin cycle. And then there are other plants as well. They're called CAM plants, which stands for crassulacean acid metabolism. अब ये जो है ना, they're mostly desert plants, जैसे कि cactus. And अच्छा हाँ, इसकी examples भी you should remember. दोनों की examples are very important. So what they do is कि they open their stomata at night. ये दिन में खोलते ही नहीं है. And then they incorporate CO2 into organic acids, which are stored in mesophyll cells. During the day, the stomata close, and the CO2 is released from the organic acids for use in the Calvin cycle. यहाँ पे bundle sheath cells का कोई चक्कर नहीं है. They have a very simple solution. ये लोग सिर्फ रात में स्टोमेटा खोलेंगे, दिन में खोलेंगे ही नहीं. जब दिन में बंद रहेंगे तो CO2 दिन में आएगी ही नहीं. तो रात के वक्त CO2 अंदर आएगी, and then the CO2 will react with organic acids, and organic acids के साथ react करके it will be stored as a compound. ठीक है, जब दिन में स्टोमेटा बंद होंगे तो वो वही रिलीज कर देंगे. CO2 will be released, और वही CO2 जाके कैल्विन साइकिल में एंटर होके it will form carbohydrates as usual. बट यहाँ पे उसने फिजिकली सेपरेट किया हुआ है. So this is like more details of the C4 pathway. You have an enzyme called PEP carboxylase phosphoenol pyruvate. Remember that. Moves into bundle sheath cells. CO2 is released and enters the Calvin cycle. Okay. Just make sure you learn their examples. Again, this is like another explanation. Any questions? Does anyone have any questions so far? Okay. So the next important chunk that we have to cover is cellular respiration. It is simply a process in which food molecules are broken down to release energy through a series of redox reactions. It's going backward. Okay. So यहाँ पे we're doing the exact opposite of photosynthesis. We're taking our food and we're using oxygen to break it down into carbon dioxide and water and energy. Like we want our energy from food. वहाँ पे we were using energy to make food, और यहाँ पे we're utilizing, we're trying to release energy from food. So respiration is of two types, aerobic and anaerobic. Aerobic में we consume oxygen, and anaerobic में we don't. Now for to complete the process of cellular respiration, ठीक है, इसमें there is an aerobic component as well as an anaerobic component. and glycolysis is the anaerobic component क्योंकि इसमें we are not utilizing oxygen. And the thing to remember is that it takes place in the cytosol. It is the anaerobic phase क्योंकि glycolysis में कोई oxygen use नहीं हो रही. And glucose is converted. Glucose is a 6-carbon compound. It is converted to pyruvate, which is a 3-carbon compound. And net ATP produced, it's 2 molecules of ATP. And then you will also have 2 NADH and H plus. Yeah, as mentioned here. So in glycolysis के कुछ initial steps में we will have to expend some energy. ठीक है? Energy is actually used. Is someone waiting to join? So energy is used in the first few steps of glycolysis. It is a 10-step pathway. Okay, there are like 10 steps in total. And then there's an energy payoff phase. Yahan pe we will see that ATP is finally formed. And then yahan pe there's the net conversion. So these are the steps. We start with glucose and we consume ATP to convert it to glucose 6-phosphate. तो ATP का जो phosphate है वो जाके glucose के साथ attach हो जाता है and that is called phosphorylation. Then in the next step, the glucose 6-phosphate is actually, this is isomerization. Okay? क्योंकि glucose and fructose they're isomers. तो glucose यहाँ पे सिर्फ एक simple सी rearrangement होती है, so that is isomerization, and you get fructose 6-phosphate. Now in the third step, you use another ATP molecule and you phosphorylate fructose 6-phosphate into fructose 1,6-bisphosphate. Now यहाँ पे something interesting will happen. यहाँ पे this 6-carbon compound, it is split into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Now this is the same compound जो उसका end product था Calvin cycle का, लेकिन यहाँ पे वो end product नहीं है, यहाँ पे it is an intermediate. It will be converted further into bisphosphoglycerate. मतलब यहाँ पे reverse हो रहा है, जो Calvin cycle में हो रहा था ये उसका reverse process हो रहा है. Glyceraldehyde 3-phosphate अब यहाँ पे जाके it will be oxidized or reduced. Can someone tell me in the chat? Is it 10 or 11 steps? मैंने अभी पढ़ा था somewhere that was 10 steps. What does this say? Glycolysis में अच्छा यहाँ पे इन्होंने total number of steps नहीं दिया हुआ. So for the number of steps in glycolysis, यहाँ पे it says 10 steps. Punjab book में it says 10 steps. So you should verify this. क्या लिखा हुआ है? Check from whatever book you're following. This says 10. तो मैंने यहाँ पे 10 mention किया है. हाँ, तो यहाँ पे is glyceraldehyde 3-phosphate, is it being reduced or is it being oxidized? Oxidized, yes. Sindh में it's 11, then you do 11. Yeah, so it is being oxidized क्योंकि यहाँ पे ये अपने electrons lose करेगा. ये electrons lose करेगा, NAD plus accept कर लेगा. अब ये जो है ना, ये electron carrier है. NAD plus is the electron carrier of respiration. NADP plus was the electron carrier of photosynthesis and NAD plus is the electron carrier of respiration. ये यहाँ से इलेक्ट्रॉन्स पकड़ेगा और ये NADH में कन्वर्ट हो जाएगा. And NADH is actually a very useless form. I'll tell you why in a minute. तो G3P एंड डाइहाइड्रोक्सीएसीटोन फॉस्फेट, ये दोनों भी आइसोमर्स होते हैं और ये आपस में इंटर कन्वर्ट होते रहते हैं. Oxidation followed by phosphorylation. Again, oxidation followed by phosphorylation produces two NADH molecules and two molecules of bisphosphoglycerate, each with one high energy phosphate bond. Okay. Removal of high energy phosphate by two ADP molecules. अब यहाँ पे ये 1,3 बिस्फॉस्फोग्लिसरेट है ना, अब इनका एक फॉस्फेट निकल आएगा और वो फॉस्फेट जाके ADP पे अटैच होगा एंड देन in this way ATP will be formed. तो ADP will be converted. ये यहाँ से phosphate उठाएगा यहाँ से and it will become ATP. Likewise, ADP will be converted to ATP. So you get two molecules of ATP. Actually, you get you get two molecules इधर. And then removal of high energy phosphate ये हो गया. Removal of water yields to phosphoenol pyruvate. यहाँ पे what is happening is I think they missed this step. यहाँ पे isomerization हो रही है फिर से, क्योंकि ये जो 3 वाला फॉर्म है ना, यहाँ पे फॉस्फेट थर्ड पोजीशन से जाके निकल के सेकंड पे लग जाएगा. So the position of phosphate shifts. So 3 phosphoglycerate will be converted to 2 phosphoglycerate. They're both isomers. And then 2 phosphoglycerate में से एक water molecule निकल जाएगा and you'll get something called phosphoenol pyruvate. And then finally phosphoenol pyruvate का phosphate निकल जाएगा and then you will get ATP in this way. तो यहाँ पे भी ATP बन रहा है, वहाँ पे भी बन रहा है. So in total that is 4 ATP molecules being produced. So 4 ATP produced, but remember that we used up 2 ATPs in the beginning. तो फिर हमारा जो net production होगा 2 ATPs net. You are unlikely to be asked these intermediates बीच में कि कौन सा compound कहाँ बन रहा है, क्या बन रहा है. But what you will be asked definitely is this कि कितना ATP बन रहा है and glycolysis कहाँ हो रहा है, what is the electron acceptor and how many steps. इस तरह के questions ज़रूर आएंगे. So far, does anyone have any problems understanding this? Net gain is of 2 ATPs, exactly. Does anyone have any problems understanding this? ठीक है. So that's glycolysis done. So I said कि NADH is very useless. And it is useless क्योंकि अब हमारे पास NADH ही रह गया है बचा हुआ. इलेक्ट्रॉन कैरियर हमारा ख़त्म हो गया है. हमें इलेक्ट्रॉन कैरियर दोबारा इसको रीजेनरेट करना पड़ेगा. We need NAD+. We need NAD+ so that we can use it over and over again to keep the process of glycolysis going. So let's move on. अब now end result इसका ये हुआ कि we got 2 molecules of pyruvate from glucose. Yes, कि हमें 2 molecules मिले. अभी तक हमारा aerobic respiration complete नहीं हुआ है because 2 ATPs is not enough for a cell. It needs more than 2 ATPs. It needs loads of ATPs. तो अब हम pyruvate को भी further कराएंगे react. So the pyruvates do not directly take part in the Krebs cycle, but they first undergo transitory reactions. Transitory reactions during which pyruvic acid undergoes decarboxylation. This is what happens to pyruvic acid, and it is also oxidized. During decarboxylation, it will give off a molecule of CO2. अब ये 3 कार्बन कंपाउंड है. 3 कार्बन कंपाउंड से ये पहले 2 कार्बन कंपाउंड बन जाएगा. एसिटाइल को ए बनेगा. एसिटाइल ग्रुप बनेगा. एसिटाइल में को ए अटैच कर देंगे तो यू विल गेट एन एसिटाइल को ए. ठीक है. ऑक्सीडेशन भी होगी. ऑक्सीडेशन कैसे होगी? कि पायरुविक एसिड अपने इलेक्ट्रॉन्स दे के फिर NAD को दे देगा, एक और NADH बन गया यहाँ पे. So you see our electron carrier is being used up again and again. The acetyl-CoA now enters the Krebs cycle. Now it is ready to enter the Krebs cycle. So in this way, acetyl-CoA forms a link between glycolysis and Krebs cycle. I think this is very easy to understand. Any questions from the transitory reaction? या link reaction भी कहते हैं इसे. Any questions? Sorry, I'll write that down. Link reactions. So known as link reaction. अब आएगा Krebs cycle. So it is a series of cyclic reactions. It actually has three names. It is called the Krebs cycle. It is also called the citric acid cycle, and it is also called the TCA cycle. अच्छा, I think glycolysis का भी एक और नाम है. It is called the EMP pathway. Has anyone heard? कहीं दिया हुआ था. I think either in Punjab or the federal book. पंजाब में नहीं, फेडरल में दिया हुआ है. EMP pathway, right? So that's another name to remember if you're taking the exam in Islamabad. Also called EMP pathway. मैं नोट्स में फुल फॉर्म ऐड कर दूंगी इसका क्या है EMP pathway का later. अभी I just remembered. So yeah. So the reactions of the Krebs cycle will take place in the presence of oxygen, and they occur in the matrix of the mitochondria. So glycolysis took place in the cytosol. Glycolysis में NADH was formed, ATP was formed, and now the pyruvic acid will move into the mitochondria so that it can enter the Krebs cycle. During the Krebs cycle, various substrates give off 2 molecules of CO2. It has 8 steps, each catalyzed by a specific enzyme. The enzymes you do not need to know. Let's move on to the chart. Yeah. So, the first compound in the pathway in the Krebs cycle is oxaloacetate. Now you need to remember the substrates. तो इसका एक mnemonic है. I'm sure the notes will have the mnemonic somewhere. कोई ना कोई होगा इसमें. तो glycolysis चल रहा है. Okay. So it's a good one. All colleges in Kashmir shall start from March onwards. यहाँ पे the first step is actually the first substrate in the pathway is actually end में acetyl-CoA से start किया. Acetyl-CoA citrate, isocitrate, alpha ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. Now the thing with the Krebs cycle is that it is a cycle, and this cycle is actually the cycle of forgetting. You learn the Krebs cycle and then you forget it, and then you learn the Krebs cycle and then you forget it. So इसीलिए you need the mnemonic, otherwise you will not be able to remember it. So all colleges in Kashmir shall start from March onwards. उससे चलते हैं. All colleges, acetyl-CoA. Confusing हो जाएगा. It will combine with oxaloacetate, which is a 4-carbon compound. Now remember that acetyl-CoA is 2 carbon. ये oxaloacetate के साथ combine करके it will form a 6-carbon compound, which is citrate. And then citrate will undergo a rearrangement and it will become isocitrate. अब अगर ये iso बन रहा है, so you can understand कि यहाँ पे simply कोई ना कोई सी isomerization हो रही है. एक water molecule निकला, लेकिन ये water molecule आके दोबारा इसी के अंदर आ गया, तो no change at all. Pass. Isocitrate is oxidized or reduced? Can anyone tell me in the chat? Oxidized or reduced? Reduced. Oxidized. People look confused इसमें. अच्छा. देखो NAD plus is actually accepting an electron from isocitrate. Isocitrate अपना electron दे रहा है. देने का मतलब तो oxidation हुआ ना. It is being oxidized. Isocitrate is oxidized, not reduced. Whoever is saying reduced, reduction नहीं हो रही, अब oxidation हो रही है. We're calling respiration an oxidative process. Respiration है ही oxidation of food. So it cannot be reduction. Is that clear? Good. So isocitrate is oxidized to alpha ketoglutarate, which is also oxidized to succinyl CoA. पहले succinyl बनता है, हाँ succinyl CoA, क्योंकि coenzyme A भी आ जाता है. So you get succinyl CoA, and then succinyl CoA is converted into succinate. ठीक है, तो coenzyme A निकल गया बाहर. अब यहाँ पे you are getting your first ATP. You're getting your first ATP of the Krebs cycle, and this is substrate level phosphorylation. I hope you know what phosphorylation is. It is simply the addition of a phosphate group ठीक है. सबस्ट्रेट लेवल क्यों हो रही है? क्योंकि सक्सिनिल को एक सबस्ट्रेट है, ठीक है, सक्सिनेट भी एक सबस्ट्रेट है. यहाँ पे सिंपल एक कंपाउंड का फॉस्फेट उठाया और जाके एडीपी पे लगा दिया. ठीक है, यू टेक अ फॉस्फेट ग्रुप, from a substrate and hand it over to ADP. So this is substrate level phosphorylation. Glycolysis में भी substrate level phosphorylation हो रही थी in these steps. ये वाले steps में सारे. So this is all substrate level phosphorylation. Substrate level. And photosynthesis में photophosphorylation हो रही थी. We were using the energy of light to synthesize ATP. To carry out this phosphorylation, हमें light की ज़रूरत थी, which is why we were calling it photophosphorylation. And this is the only substrate level phosphorylation वाला step in the Krebs cycle. Okay. So succinate is converted into fumarate. अब यहाँ पे एक और electron carrier आ गया. This electron carrier is called FAD. It is a lot similar to NAD. ठीक है, except for one difference जो electron transport chain में नजर आएगा. Then fumarate is converted into malate. यहाँ पे no ATP, no NAD involved. फिर the malate is again converted to oxaloacetate, and यहाँ पे malate is oxidized. Malate is oxidized to oxaloacetate, and you have, इसको actually color code कर लेते हैं ताकि end में calculation would be easier. What just happened? Okay, so the power went out and kids in my apartment have gone crazy. NADH मैंने हाईलाइट कर लिया, कलर कोड कर रही हूँ मैं सिर्फ ताकि एंड में आसानी हो. NADH, that's done. FAD इधर हो गया, ATP को भी कर लेते हैं. ATP, that's the only ATP we get here. I hope you all are following me so far. Great. for the formation of ATP. Does anyone have any questions? 1 NADPH, 3 ATP. That's the difference. That's the only difference between them. दोनों electron carriers हैं. NAD भी electron carrier है, FAD भी electron carrier है. But remember that NADH, अच्छा, you made a mistake here, Hamza Muin. ये NADPH नहीं है, ये NADH है. That's what I was saying जब से कि photosynthesis का electron carrier कुछ और है और उसका electron carrier कुछ और है. Respiration का electron carrier कुछ और है. They can confuse you in the exam इस तरह से. और exam stress में एक गलत वाला टिक करके आ गए, so you lose a mark. ठीक है. ओके तो क्रेब्स साइकिल का एंड प्रोडक्ट क्या हुआ? एंड प्रोडक्ट कुछ भी नहीं हुआ, इट्स जस्ट अ साइकिल. ठीक है, इट कीप्स रनिंग. व्हाट वी गेट इज दीज NADH मॉलिक्यूल्स एंड दीज FADH मॉलिक्यूल्स, जिन जो इलेक्ट्रॉन्स अब खा के बैठ गए और इनसे हमें इलेक्ट्रॉन्स दोबारा निकलवाने, क्योंकि हमें ATP बनाना है बहुत सारा. ठीक है. नाउ अब ये इट्स लाइक यू नो दीज, ऐसे इमेजिन दीज लिटिल मॉन्स्टर्स, अजीब तरह के. ठीक है, इन लोगों ने इलेक्ट्रॉन्स बहुत सारे खा लिए हैं. नाउ देयर फुल. वी नीड द इलेक्ट्रॉन्स बैक फ्रॉम देम ताकि हम अपना काम निकलवा सकें, विच इज टू सिंथेसाइज ATP. Because without the electrons and without the ATP, the entire process is useless. So the electron transport system of mitochondria consists of a series of electron carriers which are located on the inner membrane of mitochondria. Now, please remember that it's the inner membrane and not the outer membrane. It's a highlight, सीधा नहीं हो रहा आज. During breakdown of a glucose molecule in aerobic respiration, there are 6 steps. Now forget this. There are hydrogen ion. ये वही वाले जहाँ पे जहाँ बेसिकली हमारे इलेक्ट्रॉन कैरियर्स रिड्यूस हो रहे हैं ना एंड बिकमिंग NADH, वहाँ पे we see that hydrogen atoms are also released. See, let me show you. H plus निकल रहा है, again H plus, H plus. तो यहाँ पे hydrogen ions are released. A pair of hydrogen atoms will dissociate. You can forget this. Not very important for questions. ठीक है. So now there's a chain of electron carriers. जैसे वहाँ पे electron carriers से photosynthesis में यहाँ पे भी हैं. Cytochrome B, cytochrome C, cytochrome A, cytochrome A3. Now I know कि in the federal book it is given in significant detail. ठीक है. And if I go into that detail तो बहुत ही ज़्यादा लंबा हो जाएगा. What I can do is, because it's given in your book, I can share a short video from YouTube explaining it, and I can add a supplement at the end of the notes so that it's easier for you to understand. Like, as far as the Sindh and Punjab people are concerned, that's all you need to know. Cytochrome B, say C, say A, say A3. Just remember the order B, C, A, A3. And just know कि FADH2 जब enter होगा electron transport में, तो you will get 2 ATP molecules as a result, और एक NADH जब एंटर होगा साइकिल में तो यू विल गेट 3 ATP molecules as a result. तो FAD आपको देगा 2 ATP और NADH आपको देगा 3 ATP. Just remember that. And अभी substrate level phosphorylation मैंने बताया था कि there isn't any electron transfer इसमें, because what you're doing is you're just taking one phosphate group and उठा के आपने ADP को दे दिया. There wasn't any electron transfer. Oxidative में you have the oxidation of there's oxidation of the various intermediates. ATP molecules are synthesized by using energy from oxidation of glucose molecules during aerobic respiration. So this is basically the electron transport chain. Short. Reactions of the Krebs cycle. हाँ, अब हमें अपना बैलेंस शीट देखनी है ATP की. We know that we got two molecules of ATP from glycolysis. ठीक है. अब उस 2 ATP, वो obviously 2 ATP enough नहीं है, which is why we're doing this entire thing of the Krebs cycle and the electron transport. अब notice के Krebs cycle का कितना hype था. Remember कि you know it's going to be this one big reaction. हमें चाहिए ये aerobic respiration के लिए, लेकिन वहाँ से भी we just got one measly ATP. ठीक है, 1 ATP again. सो दैट टेल्स यू द इम्पोर्टेंस ऑफ ऑक्सीडेटिव फॉस्फोराइलेशन, कि अभी तक हम सिर्फ 3 ATP बना पाए हैं, इतना करने के बाद भी। तो दिस इज द स्टार ऑफ द शो, ठीक है। सारा काम ही इसी का है, यू नो, द इलेक्ट्रॉन ट्रांसपोर्ट चेन, एंड दिस इज व्हाई द इलेक्ट्रॉन कैरियर्स आर इम्पोर्टेंट। सो लेट्स डू दिस डिटेल इसकी। सबस्ट्रेट लेवल फॉस्फोराइलेशन हुई तो वी गॉट 2 ATPs डायरेक्टली। यहाँ पे हमें 2 NADH मिले थे, रिमेंबर, फ्रॉम ग्लाइकोलिसिस। ट्रांजिटरी रिएक्शन हुआ, हमने फिर 2 NADH निकल गए। Krebs cycle में we got 6 NADH and we got 2 FADH molecules. So yeah, अब we know कि हमें एक NADH से हमें 3 ATP मिलने चाहिए, right? And according to this it should be 6. 6 generate होते हैं, but 2 of these are consumed when we are transferring the NADH to the mitochondria. जो NADH glycolysis में बने थे वो हमें माइटोकॉन्ड्रिया तक ट्रांसफर करने में ही 2 ATP खर्च हो गए, विच इज़ वाई दिस सेज 4 ATPs, ठीक है। एंड देन यहाँ से 6 ATPs बने इलेक्ट्रॉन ट्रांसपोर्ट चेन में जब NADH गया, तो यहाँ पे वी गॉट 18 ATPs, एंड इफ यू डू द मैथ्स इट अमाउंट्स टू अ टोटल ऑफ 36. Sometimes it is said 38, लेकिन 36 is the correct number. I think सारी बुक्स में unanimously 36 ही लिखा हुआ है. So make sure you pick the right number. Except in prokaryotes. ठीक है. Prokaryotes में, अच्छा, any guesses as to why 38 in prokaryotes and 36 in eukaryotes? First, let me know if all of this was clear. Which of these steps altogether are taking place in the mitochondrial matrix? All except glycolysis. Glycolysis is happening in the cytosol. सही है. And जो transitory reaction है ना हमारा, wait a second. Transitory reaction भी हमारा, actually I'm not very sure about this. So let me check. I'll check कि transitory reaction कहाँ होता है. See, glycolysis, this happens in cytosol. ये कहाँ होता है? I'll check and let you know. I will check. Krebs cycle happens in the mitochondria. यही 3 steps है ना, इन्हीं की location है. Obviously जो हमारा oxidate जो electron transport chain है, that is also in the mitochondria. Matrix के अंदर Krebs cycle होता है. This happens in the membrane, inner membrane. Clear? ट्रांजिटरी रिएक्शन सिर्फ ये करता है कि पायरुवेट को कन्वर्ट कर देता है टू एसिटिल को ए. Gets inside the membrane. What are you asking a question, or are you you're welcome. Gets inside the membrane. I'm not sure. I could, if you're trying to ask something, so please rephrase. अच्छा, तो ये तो पूरा मैथमेटिकल कैलकुलेशन हो गई कि कौन से स्टेप में कितना ATP बना, कितना NADH से कितना ATP बना, एंड आई थिंक दैट इज इट. इसमें रेस्पिरेशन में फर्मेंटेशन रह गया. वेट, when pyruvate travels to the membrane of the mitochondria, is that the transitory reaction? ये traveling नहीं है transitory reaction. Transitory reaction is simply the conversion of pyruvate into a form that can enter the Krebs cycle, ठीक है? Because pyruvate cannot directly enter the Krebs cycle. It has to be converted to something, and that something is acetyl-CoA. Does that make sense to you? हाँ, वो ट्रैवल जरूर कर रहा है, लेकिन वो ट्रैवलिंग नहीं है ट्रांजिटरी रिएक्शन. दैट्स नॉट द पॉइंट. Transitory reaction is simply its conversion into acetyl CoA. इसको लिंक रिएक्शन भी कहते हैं. बस इसका ये है कॉन्सेप्ट. एंड आई होप ऑल ऑफ यू अंडरस्टैंड कि 36 ATPs are generated. अच्छा, so I mentioned कि 38 ATPs बनते हैं in prokaryotes like bacteria. Can anyone guess why? उनको उसको 2 extra ATP कैसे मिल रहे हैं? हम like we, you know, as eukaryotes, they're more advanced. उनको ATP ज़्यादा मिलना चाहिए. No transitory reaction. या ट्रू. बिकॉज वहाँ पे मेम्ब्रेन बाउंड ऑर्गेनेल्स नहीं है, वहाँ पे कोई भी चीज़ कहीं से इधर उधर ट्रांसपोर्ट करने की ऐसा ज़रूरत नहीं है. तो जब ट्रांसपोर्ट नहीं हुआ, पायरुवेट इधर लाइक एक जगह से उठ के कहीं दूसरी जगह नहीं गया, तो फिर व्हाट्स द पॉइंट? व्हाई डू द ट्रांजिटरी रिएक्शन? व्हाई मूव द NADH एट ऑल? जब वो मूव नहीं हुआ तो वो यूटिलाइज नहीं हुआ, तो उसको अपना पूरा ATP मिला. So that's just food for thought. Exactly. No, in prokaryotes, no ATP is used in transportation. So now this was aerobic respiration, and I know all of you know कि there are conditions जहाँ पे aerobic respiration becomes difficult या जहाँ पे aerobic respiration नहीं हो पा रही होती, such as in active muscle cells, में for instance. तो अब what happens is, अब एरोबिक रेस्पिरेशन अगर नहीं होगी तो इसका मतलब है कि हमारे पास इलेक्ट्रॉन ट्रांसपोर्ट चेन नहीं हो पाएगी, बिकॉज यू नीड ऑक्सीजन फॉर दिस. एंड व्हाई डू यू नीड ऑक्सीजन? क्योंकि इलेक्ट्रॉन ट्रांसपोर्ट चेन में जो फाइनल इलेक्ट्रॉन एक्सेप्टर है, दैट इज ऑक्सीजन. इफ वी डू नॉट हैव ऑक्सीजन, तो कौन एक्सेप्ट करेगा? दिस इज आवर फाइनल इलेक्ट्रॉन एक्सेप्टर. ठीक है. एंड इफ देयर इज नो ऑक्सीजन एट ऑल, तो यहाँ पे इलेक्ट्रॉन को कौन रिसीव करेगा आके? तो इलेक्ट्रॉन ट्रांसपोर्ट चेन हो ही नहीं पाएगी. जब इलेक्ट्रॉन ट्रांसपोर्ट चेन नहीं होगी, तो ये जो NADH है और FADH है ना, दे आर इंटायरली यूजलेस, बिकॉज वी कैन नॉट गेन ATP फ्रॉम देम. हमारे 36 ATP नहीं बन पाएंगे फिर. वील जस्ट बी स्टक विद द 2 ATPs दैट वी गेट इन ग्लाइकोलिसिस, एंड वील जस्ट बी स्टक विद पायरुविक एसिड. और वो जो 2 ATPs होंगे, उन्हीं से काम चलाना पड़ेगा. And that's not a nice thing for our cells. The cells would die for lack of energy. So इसीलिए we have anaerobic respiration as well. And respiration in the absence of free atmospheric oxygen is called anaerobic respiration. It is also called fermentation. It takes place in yeast, it takes place in bacteria, it takes place in some gut parasites, and in the muscle cells of vertebrates during vigorous exercise. The energy yield is small, just 2 ATPs per glucose molecule. The end products are either ethyl alcohol or lactic acid, ठीक है? Depending on the organism. Yeast में alcohol बनेगा, many bacteria and muscle cells produce only lactic acid. I hope that is clear. Any questions इसमें से? Clear. So fermentation is just an alternative term used for anaerobic respiration. ठीक है? It includes glycolysis. Now this is important. This is very, very important. If fermentation is the sum of glycolysis and the reduction of pyruvic acid. ठीक है. So glycolysis plus the reduction of pyruvic acid is fermentation. ठीक है. और जो आपके पास 2 ATPs बन रहे हैं ना इसमें, वो आए ही glycolysis से हैं. ये सिर्फ glycolysis से ही आएंगे. Reduction of pyruvic acid. ठीक है. Can anyone guess as to why this is important? What is not clear? Hamza Moin, why have you written nope in the chat? Acha, clear. Right. Yeah, so I was asking कि अभी इसमें लिखा हुआ है कि फर्मेंटेशन से दो एटीपीज बनेंगे, एंड जो ग्लाइकोलिसिस है उससे भी दो, ग्लाइकोलिसिस में दो एटीपीज बनेंगे, फर्मेंटेशन में दो एटीपीज मिलेंगे, लेकिन फर्मेंटेशन ऑल्सो इंक्लूड्स एन एक्स्ट्रा स्टेप, द रिडक्शन ऑफ पायरुविक एसिड. कैन समवन टेल मी व्हाई इज इट इंपॉर्टेंट टू रिड्यूस पायरुविक एसिड? एटीपी तो बन ही नहीं रहा, फिर क्या जरूरत है? लैक्टिक एसिड. हाउ डू यू थिंक लैक्टिक एसिड इज अ यूजफुल प्रोडक्ट? लैक्टिक एसिड जब जमा होता है मसल सेल्स में, ऑल वी फील इज पेन. How is that a useful end product? Why are we carrying out the reduction of pyruvic acid? Nahin, ethyl alcohol and CO2, like outside the cell. Okay, I think I'll explain. I'll explain this. Again, वही इलेक्ट्रॉन कैरियर वाली बात है. अगर हमारे पास ऑक्सीजन नहीं भी है, अगर हमारे पास ऑक्सीजन नहीं भी है, ठीक है, तो पायरुविक एसिड को रिड्यूस कौन करेगा आगे? एनी गेसेस टू दैट? कौन रिड्यूस करेगा पायरुविक एसिड्स को, पायरुविक एसिड को? आई हैव टॉक्ड अ लॉट अबाउट दिस during the past 2 hours or so. There is one compound that is constantly, being making an entrance. जब से the electron carriers, NAD plus, right? NAD plus हमारा इलेक्ट्रॉन कैरियर है, और जब वो कन्वर्ट हो जाता है NADH में, जब वो रिड्यूस हो जाता है इलेक्ट्रॉन खा के, तो इट बिकम्स यूजलेस, है ना? तो NADH हमारे पास जमा पड़ा हुआ है इतना सारा, ATP हमारे पास सिर्फ दो ही बना है. अगर सारा NADH बन बन के यूजलेस होता रहेगा, तो हाउ विल द प्रोसेस ऑफ ग्लाइकोलिसिस गो ऑन? We know that the cell does not have oxygen. We know that the cell needs to do anaerobic respiration. लेकिन ग्लाइकोलिसिस करने के लिए भी आपको NAD प्लस चाहिए. फ्री NAD प्लस खत्म हुआ जा रहा है बिकॉज इट्स बीइंग कन्वर्टेड टू NADH. To get your NAD plus back, you need to convert it to NADH. Sorry, you need to convert it to NAD. You need to convert it to NAD plus. ठीक है, NADH को आपको convert करना है इसमें. और ये conversion कैसे होगा? जब ये अपना एक इलेक्ट्रॉन दे देगा वापस, जब वो एक इलेक्ट्रॉन अपना पायरुविक एसिड को देगा, तो पायरुविक एसिड रिड्यूस हो जाएगा, आपको NAD प्लस मिल जाएगा दोबारा. इज दैट क्लियर? दिस इज द इंपॉर्टेंस ऑफ फर्मेंटेशन. दिस इज वाई, अगर इफ anaerobic respiration क्यों हो रही है, this is why it's happening. And pyruvic acid, I'll explain this in a new page. ठीक है? Look, we have pyruvic acid as the end product. Pyruvic acid from glycolysis. ठीक है? And we have our two ATPs. वो दो ATP जो हमें काफ़ी नहीं है for the cell to function. But फिर भी हम काम चला रहे हैं because we don't have a choice. ठीक है? We have our pyruvic acid from glycolysis. And we also have loads of NADH. Useless because we don't have our electron carriers. ठीक है? ये अब हमें इसको रीजेनरेट करना है. We need to regenerate. We need to regenerate NAD plus in order to keep glycolysis running. For this regeneration, we reduce, मतलब give electrons to N pyruvic acid. Okay. So what pyruvic acid will do is pyruvic acid plus NADH will give you something. It will give you a product, the product of fermentation, and it will give you your electron carrier back. हमें इसी से गर्ज है बस, ये वापस चाहिए. ये दो तरीकों से वापस आ सकता है, या तो अल्कोहलिक फर्मेंटेशन से आएगा या लैक्टिक एसिड फर्मेंटेशन से आएगा. लेट्स स्टडी बोथ. ठीक है. यहाँ पे pyruvic acid alcoholic fermentation, which is what happens in yeast. Pyruvic acid is converted to acetaldehyde and CO2. Again, ये learn करने वाली चीज़ है बस. And then acetaldehyde plus NADH will give you ethyl alcohol and NAD plus. अब यहाँ पे क्या हो रहा है? This is simply the reduction. Just happened. This is the reduction of pyruvate. And this is also the reduction of pyruvate. I hope that is clear. When will the class end? It's about to end. बस हो गया. We actually had some time left, तो मैंने थोड़ा सा time लगा के ये fermentation भी बता दिया. Is that clear? Is everything clear so far? Okay, I'm glad. ठीक है. And chemiosmosis. Okay. अब जिस तरह से chemiosmosis was being used to synthesize ATPs in photosynthesis, इसी तरह से there is a similar process here, and I think you should read it on your own because the pattern doesn't change. There are a lot of similarities, and that's it. That's it for today for this class. I'll assign you questions. When you'll do more questions, so hopefully these things will be clearer. And also, good luck for your AKU test, whoever is sitting for the test tomorrow. Make sure you sleep well. I don't know how many of you are also sitting for the test tomorrow. I think strength is really come. Okay. I'll end the meeting.